Okay, thank you for the introduction by Dr. Fan. So today I'm really focusing on what I'm doing for the last 2 years, and uh, I will be the main presenter today, but my colleague, Dr. Mingyu Yang, she will also be around to answer any questions you may have for the data analysis for our DBiT-seq. Um, let's get started. So with really good review sections done by both Dr. Coskun and Dr. Fan, I think I don't need to spend too much time on the background technology introduction here, but I do want to emphasize some of the next generation sequencing-based technology here. So the first one of this technology was developed at year of 2016, and at that stage, it's really a new technique and clever method to really look into expression patterns in tissue. So before that, all the technology was done by basically micro dissection, basically need to really dissect the tissue into small pieces. Then you can sequence them such as they are like the single cell RNA sequencing process. From here, we don't need to really dissect the tissue anymore. We just can put the whole piece of tissue on top of a arrayed probe capture spots. So those spots will be really acting as like hotspots. You can really retrieve those spatial information after you did the whole process and get the sequencing data for each of the spots. So the process is simple. They just place the tissue on top of the spots array. Then you have this spatial barcode. Then by sequencing this barcode, you can retrieve back the information of each spot. But at this stage, I think this technology already adopted by the 10x Genomics. They use their wisdom technology basically based on this technique about 2016, but they got better resolution now, around 55 micrometer. A second technology I want to introduce here is we call slide-seq technology, about 3 years later after this 2016 technology. So basically the whole process is quite similar, but they have a different way to prepare those spot arrays. So in order to reduce the spot size, I think, which is pretty hard for spotting technologies nowadays, they use beads. So basically beads, you can really control the size of the beads by choosing different size of beads. Here they use 10 micron. I think that will be optimized for, there will be a balance between how much probes you can have on the beads and also the resolution, I think. What they do, they just put the beads compactly assembled on the glass slide, then they will sequence the beads beforehand, then just place the tissue on top of those beads, then get the tissue process, and then later use the beads information, they can retrieve back the spatial information of the tissue. So with this technology, they can improve the resolution to 10 micron. And a third one we have here is really, really high resolution. So it's like 2 micron a size, and which could be considered as a sub-cell resolution. The whole idea is also similar, but they are using much smaller bead size. Here it's a 2 micron. So, but that will be it's kind of because they use much smaller size, they kind of sacrifice the quality of the sequencing because the beads are too small that you don't have enough beads on it. So I think all the 3 technologies here, they are based on idea that you really need to do things outside of the tissue, because your tissue is on top of the spotted slide, and your probes are on the slide, but you really need the RNA inside the tissue to be diffused out, so that can be captured by the probe spots underneath. So I call this like all of cell barcoding methods because you really need the diffusion process to make it work. So that could be problematic in some ways. For example, when you place your tissue on top of your spot array, so you need to be really careful if you place that. If the tissue has some wrinkle on it, then you need to really redo it. Probably that process may damage your underlying slides. And also, they really need high demanding technology for slide preparation. For phase 2, when you place the tissue on top of the slides, then you need to rely on the diffusion process. Namely, that you need the RNA to be diffused out of the tissue and bound to the probes. But if you're doing that way, you cannot really make this work with any formaldehyde fixation process, which is very popular in this field. And that means for this current method, you can now work on FFPE samples because most of them are just fixed by PFA beforehand. And also, it could be hard for them to incorporate other omics like proteomics because they rely on the diffusion process. If you have a protein here, protein is pretty large. They can hardly, I think, diffuse fast into out of the tissue. So the question come here is like, can we do in-cell barcoding rather than out-of-cell barcoding? So I think the idea has been brought out about more than 20 years ago. So basically, groups demonstrate that you can really do an in-cell RT process, produce cDNA inside the cell, and do a RT-PCR also inside the cell. So basically, the cell will serve as a reaction container. You can have the RT reaction happening there, and also the cDNA will be just trapped in the cell, and you can further process the cell as a unit. Yeah. So this idea also being adopted by the ICI RNA-seq method on 2017. Basically, they just doing it's just use a cell as a container doing an RT reaction. And also later they can do the retrieve the cell cDNA, then doing pooling, then do the sequencing method. Similar split-seq also using this method. So which means that we can really doing an in-cell RT process. So it makes the in-cell barcoding possible. And another question like, because it's hard to be adopted for current technology to do the proteomics, but for single-cell RNA-seq, protein analysis were made possible by so-called STAT-seq process. So I think it's a pretty smart idea that you just need to incubate, just pre-incubate your cell with oligo labeled antibody, and then from then on, you are ready to go with any droplet-based sequencing process. So your cells are definitely the same as before. It's just like you have more information with the surface antigen here. So the whole process is similar as DBiT-seq or 10x genomic sequencing method. But you can see from the right here that with the addition of protein or antibody into it, you can really do a much better or improved classification of those PBMC cells because surface proteins are more unique features for those cells. Okay, another thing I want to mention here is so-called the two-dimensional barcoding process. Because our tissue is kind of like a two-dimensional object here. If you want to find information of the tissue, especially the spatial information, what I can think of just need to really split them into small squares. As shown here, like you have a 2D barcode, you have x-axis and y-axis. With the x and y location information, you can really find the spot here. So this is what we use for our method. So for example, you have your tissue, and if you really put x and y coordinates inside the tissue, you can later retrieve back the spatial information. So in this way, we what we do is we use the microfluidic device to achieve that. Microfluidic device have been long ago used as being popular in this field, and our lab have been developing those technologies. And here shows a really PDMS stamp method. We can just doing a high density DNA barcode by simply flow the DNA solution into it and drag it up. So this film we will fill where strips of DNA capturing spots. Then you can just capture the target DNA. All you can do with here is the antibody based barcode chip, so that you can really capture either immune cell release cytokines, or you can just capture any blood proteins. So then I want to move on to the workflow of our technology here. So basically, you can start with a tissue slide. You can stain it with any immunofluorescent dyes or antibodies or DBiT, whatever you want to find out the morphological information of your slide. Then you can doing a fixation. Basically, just use a very common formaldehyde fixation at room temperature for 10 minutes. We do want the tissue slide to be kind of thin, so less than 10 micron. Then you can also do the immunostaining if needed. Then you can move on if you want to do the protein analysis or proteomics along. You just need to add the antibody-derived DNA tag into it. So I think it all depends on the mixture you want to, how many antibodies you want to do here. If you're doing over 100, it's still possible. I think use our method. And after that, I think your tissue is ready to be processed by our technology. You just need first PDMS, which have 50 parallel channels. Then you can flow in the Barcode A solution along with reverse transcriptase. So that will do RT process in 2 hours. So later you just clean with PBS by flowing it through. Then follow that, we just inject the ligation solution, which you have DNA ligase along with the second barcode. So the second barcode will be lysed to the first barcode. Then you can have information about the second dimension. So incubate for another 30 minutes, just to clean up. Then you can, I think the tissue is ready to be either imaged or being lysed, then collect into a tube, then doing all the things you do for quite like a single cell RNA-seq. So the imaging here, you don't really need a very high resolution microscope. Any light microscope will work because you just need to find out the tissue location and maybe a channel on top to help you later identify the location. And the digestion is pretty standard, just proteinase K digestion. You collect them into a tube, then PCR and build library. So a little bit about the chemistry. And chemistry is also, I think, simple to understand. First, you have the in-cell, the mRNA, the poly-A tail with poly-A tail. Currently, we can only work on poly-A tail once, but I think our technology also can be adapted to any non-poly-A tail mRNAs. And you have the ADTs, also they are labeled with an antibody barcode along with the poly-A tail. So if you flow in our barcode A, which has a poly-T, it's kind of serve as a RT primer region, then you have the barcode A to tell the location of each channel. You have a ligation linker part. Then to the RT process, you have a cDNA with mRNA information or antibody information. Then you flow in barcode B doing ligation. So basically this ligation linker will be ligated to this ligation linker. Then you have a ligase product, which is a final product. We also have a biotin here to be used later to extract or purify this product. So basically you have a barcode A and barcode B and UI mice. So basically barcode A will tell you the y-axis information, and barcode B will tell the x-axis information. So combine these two, you have a two-dimensional barcoding of the tissue. So this is how what the device look like. I think there are mainly 3 pieces. You have a glass slide with your tissue on top. Then you put the first layer of PDMS chip on top. The PDMS chip have 50 wells serve as 50 inlets, and you also have 50 outlets. Then you have a clamp which helps to seal the PDMS on top of the tissue to avoid any possible leakage. Then you just apply a vacuum, so you just pull solution from this side to the other side. Those channels will be filled up with solution, and possibly can do RT or ligation process. So the resolution of our DBiT-seq cannot be controlled by the width of channels. So I think it's pretty convenient. So we can just have a 3 different set of designed PDMS chips. Then you can just modify mainly this area. So with this different channel width from here 10, 25, and 50, you can just control the resolution of your barcoding process. Basically, you can have tissue pixel size from 10 to 50. So with different needs. So what here I show is enlarged picture of the channel area. So basically, you have 50 parallel channels. Then each channel will be isolated from others. Then each channel will have a distinct barcode. Okay, so the validation of the DBiT-seq technology is pretty straightforward because you have basically a first direction of channels. You flow the first solution, then you have a second direction of channels. Basically, you are looking for squares. So those array of squares, each square will be a hotspot which will be later sequenced. So what you are looking will be just those squares. So square area will be where the samples will be processed later. And here we show the staining, a simple staining process, basically just flow 2 different color nucleosides. So with the first direction to be blue, the second direction to be red. So you can see a very nicely patterned tissue with the squares in the middle, and they are connected by different channels. So there are minimal leakage between the channels and the outside wall area. And another way to further validate the technology is to use fluorescent labeled barcode DNA oligos. So basically, for the first direction, we just flow a red color CY3 labeled Barcode A solution, which will just bind to the mRNA in the cell. And we just flow a second Barcode B, which will be ligated to the Barcode A. So Barcode B will have green color. So if you look at only the green color, it will just form squares, which means only the spot in the middle, only the spot with a Barcode A, and also have the Barcode B flow through, will have this square blue square with blue color. So you can see there's very little field leakage between the channels. And and here's another picture we show here is a 10 micron resolution. So basically you can see a very clear and clean square arrays of square here. And also you can see the basically the square is shown here because the walls will press down the tissues nearby, kind of seal the square. And we also validate the diffusion process. Possibly there could be leakage of minimal diffusion process. We see that the diffusion less than 1 micrometer, and also for 10 micron resolution, and about 4 microliter for 15 micron resolution channel. And the average cell number for 10 micron resolution will be about 1.7 cells, and there will be 25 cells in 15 micron. So if you want to study really a few number of cells or layer of cells in a tissue, probably just go for the 10 micron resolution. And we also validate our technology by comparing aid with previous technology like ST, which is a 2016 science paper, and also SLIDE-seq. So basically, want to emphasize here, like with the reduction from 50 micrometer to 10 micrometer, we didn't see a very dramatic decrease of the RNA being sequenced, RNA being identified. I think that's mainly because we are doing an in-cell process. We're doing the in-cell RT process, so we are not limited by the surface probe counts or surface probe densities. So we can do relatively much better capturing of the cell of the RNAs inside the cell. Similarly here, this shows the transcript counts. Another validation we do is use the single molecule FISH, because it's the golden standard for validation of these spatial technologies. So smFISH basically just design probes which can bind mRNAs, maybe multiple or tens or hundreds of those probes which can bind mRNA and show the fluorescence. So basically, we are comparing pairwise the smFISH data with our sequence data. I think they are pretty showing a similar location and patterns. So this one for TRF also showing very well. Basically, we test 4 different smFISH technology to show the the transcripts are overlaying pretty well with our technology. Then we go ahead and compare the transcript counts we got from smFISH and with DBiT-seq. So basically, smFISH kind of have higher efficiency for capturing those mRNAs. We show about, on average, 15% of total mRNA transcripts being identified by smFISH were captured by our technology, and this could be further improved if we just improve the sequencing depths. And now I want to move on to the real tissue we work on. So first thing, we just want to work a large area with a 50 micron resolution. This is a whole embryo. It's a day 10 embryo. We're staying with H&E, so it shows clearly it's a whole tissue. Then we just doing our technology and show here the Pan-mRNA, basically the total mRNA counts for each of spot, and also the total protein counts for each spot. We have a 22 protein panel here, and you can see there a little bit of the bands here, which could be caused by in humans. It's the barcoding process or flowing process now, so homogeneous between different channels. But when you look at the genes after standard normalization method used for siRNA-seq, we can see those binding are just dispaired. I think that's because normalization can just because normalization will just remove the difference between each channels and doing very well and showing no binding for those high expressed genes here. And another thing we can do is just simply seize each of the spots as a single cell. So we can apply the single cell RNA technology, the clustering technology, to our spatial data. We just say them as single cell cluster with UMAP and choose the clustering. We can then place them back on their tissue location, and overlay shows different distinct clusters. They're also showing a very meaningful overlay with annotated tissue areas. For example, here from the geo analysis, you can tell the different regions of the tissue, and we also compare them with just the H&E-based annotation method. They are matching pretty well. And another thing I want to mention here is we want to compare our data with the single cell data, because single cell data they are more based on single cells, and it seems cleaner because our spots may contain more than one cells. So we still want to compare our data with any single cell data here. So we just do a pseudo-bulk sampling of the data reported by Nature paper 2019. They're doing very nice work on single cell sequencing for different stages of the mouse embryo. And we're just doing a pseudo-bulk sampling of their tissue, basically just aggregate the expression data from single cells into a bulk tissue and just trying to align our data into them. I think because we have an E10 sample, so that lies pretty, they all lie pretty well between day 9.5 and then day 10.5 data, so which also demonstrate we are doing a quite precise measurement of the transcriptome. So another power of our technology that we can measure protein and mRNA side by side. So basically, we have 22 protein profiles. Basically, you can compare them side by side. But you can see here, like usually the mRNA expression doesn't really match pretty well with this mRNA. Sorry, this mRNA expression doesn't really match well with the protein expression. I think that's pretty due to the different because protein are more kind of stable and mRNA are more transient, so there could be difference between them. And so we also did this measurement. Basically, we just annotated the tissue and annotated the mouse embryo into 13 different areas based on their morphology. Then we just extract the proteins and also mRNA counts for different areas. Then we put them side by side. So red will be mRNA and blue will be protein. So again, it shows that the protein expression or protein counts really doesn't really reflect with the mRNA counts. I think that is kind of reasonable because as the process of transcription and process of translation are affected by different factors, and also the mRNA tends to be more easily degraded compared with protein. So the protein will tend to be more stable and the mRNA will be more transient. So there could be differences of them. So I think that's also showing the value of marrying a protein also along with mRNA. So we then trying to lower or increase the resolution by kind of zooming the mouse embryo tissue. So we focus on the brain area. So in this way, we can just increase the resolution from 15 micron to 25 micron. So the data here shows more clearly the spots. The spots are much smaller, I think. And we show here, like if you have a very distinct feature in your brain, then that can be also observed by our Pan-mRNA sequencing data, because this will be just relevant to the transcript counts. Because if you have dense cell in some area, you already have more RNA counts in that area. So we're also doing a simple clustering based on just the spot transcriptome information, and it also shows a very, I think, not very good, but still good clusters distinguish some meaningful features of the tissue. For example, the heart here, and also the midbrain area. And also we clustering use proteins. So the proteins are kind of showing more distinct clusters than mRNA. That's probably because mRNA have large background signals, but proteins are pretty clear. And we then want to explore the capability of the protein expression patterns, what you can tell from us. So we show here is a comparison with MRI imaging of developing vascular in the brain. So basically, we test this pan endothelial cell antigen, and it shows a pretty nicely network of those blood vessels in the brain. it's kind of very interesting here. And also we doing the epicam in measurement in mouse. So because the epicam is usually expressed only in ear and branchial arch. So here is where that shows this ear and also the branch arch here. So this also demonstrates we are doing the protein profiles pretty good for us to understand the protein locations. Then, of course, we need to compare with some immunofluorescence-based method. Basically, we staying with antibody labeled with fluorescence and compare with our data. So the profile here, line profile here, shows pretty good matching between the two. Similarly, we're doing this for the PEC antigen, and also it shows pretty well matching between the immunostaining data and our protein counts data. So I think another thing I want to mention here is that we can really doing a protein-based or protein-guided analysis of gene. So for example, this Medicam protein is highly aggregated in this middle brain region, and by taking cells or peak cells from this region, we can further analyze the Medicam plus cells and Medicam minus cells. So basically, it can get our understanding of the Medicam protein expression influence on their expression, RNA expression. So this kind of showing another power of technology. So then I go to the most highest resolution of our technology. It's a 10 micron resolution process. We have a 10 micron chip mainly covering the eye region of the mouse embryo. So this region about 1 millimeter by 1 millimeter, and you can see the eye, roughly the eye outline here. So the eye is just about only like 250 micron size, so pretty small. But if you see the eye development process, so it's going from starting mainly at, I think, day 7 or day 8, you start to see the optical vesicle here. You have the lens plaque here. Then you later divide into lens vesicles, which about on day 10 and on day 11, you see a pretty mature eye developed in the mouse. And we can see from here, like, is the very thin layer of the retinal pigmented epithelium cells. So these cells kind of like serve as one single layer of the cells here. And we want to show like we can see this single layer of cells by showing the by doing the mRNA sequencing. So from here you can see a TRPM1 gene. It's really showing just align very well with this pigment cells in surrounding the eye region. And we also look at the expression of other genes, which one of the interesting one is this aldehyde dehydrogenase gene. So it's showing it's being expressed as at the right part of the cell of the eyes, and pretty distinct from the other region of the cell, the other region of the eye. We don't know exactly why, but it could be very interesting biology here to be studied in the future. And this also show like we have this RPE region showing, which is a pigment cell region surrounding this area, and this will be where we can see the aldehyde dehydrogenase expression. And we also are able to compare the eye region between the E10 mouse and E11 mouse embryo. So basically E10, the TRPM1 gene is showing very good line with pigment cells, and also TRPM1 also showing good expression around the retina. And also here is another gene called MSX1. So it basically expressed in the cellular body of the eye, which includes the muscle controls of the shape of lens. So they play important roles for shaping of the eyes, because you don't want the eye to be grow without any control here. And then you have another eye closure related gene found as GATA3. So they are also showing surrounding the eye region. And I also want to emphasize here this aldehyde dehydrogenase series, the gene series, you have 1, 2, 3. So compare with some published data that it shows A2 is pretty linearly expressed in the eye region, which is shown here. And A1 and A3 are pretty expressing a lot. And A1, as I mentioned before, they are lying in the dorsal region of the eyes, which is this region, match well with our data. And for A3, so they tend to show some kind of aligning quite some alignment with pigment cells, which is shown here, and also they have some other region, which is also showing in the day 11 mouse eyes. Okay. So we also did a very quick clustering. So this non-supervised clustering. So you can see the eye being shown here, and as well as other interesting regions. So this is pretty just a good way to see the expression patterns very quick. And we adopt another technology developed on 2018. It's called Spatial DE. So what Spatial DE doing here is they really incorporate spatial information into the clustering. So previously, when we cluster, we just see each of the spots as a cell. We don't really incorporate the spatial information into it. But with Spatial DE, you can incorporate the spatial information in it. So with spatial information into it, you can make the previously kind of noisy clustering into very beautiful clustering, because you have the spatial information. The spatially closed two spots will give you more consideration when you're doing the clustering. So basically, with this process, we got a cleaner identification of different tissue features, for example, eyes, ears, and pituitary gland. So we can really, this can really help us also to identify those features which will be hard to be annotated by just H&E staining. Okay, then we just go ahead and do a analysis of all the tissue samples we have currently. We have 11 tissue samples with different developmental stages and also different resolution. You have 10, 50, and 25, and different area, the tail region or the brain region. We're just showing the whole clustering of those things, of those tissues. And also we can really doing a geo-analysis to show like the type, the different type of those clusters. Then I want to also introduce a little bit of the embryo organ development. So basically, it shows a mouse embryo development from day 0 to day 13.5. Basically, the heart start to appear at the day 6.5, and liver start to appear at day 9.5. So basically, these two organs are the earliest organs showing up in an embryo. So if you see the staining pictures, basically the heart is really here at the lower body, and also the green one is the livers. So you can also see a heart here, which is the atrium and ventricle, and clearly showing here. So we want to see if our technology can show the organ similar as the staining method. So we just cluster, look at this single tissue, which is an E11 mouse. We just covering the lower part of the body instead of the brain. So we do a simple clustering of the tissue based on the sequence data. You can see the very distinct clusters here, and we find out those clusters to be either to be liver or to be heart. And also we can see a very thin layer of distinct patterns here. So that turns out to be some skeletal muscle cells around here. And also you can see a layer a thin layer of cells around this region, which turns out to be the alta, the epithelial, the endocellular cells of the alta in the body. If you look into some special marker genes of each region, you can see the MY6 gene. So that kind of prefer expressed in the atrium instead of the ventricle. And MYL7 is kind of different. They are more expressed in ventricle instead of atrium. Then you have the neurotube specific gene PAC6, and also have a skeletal muscle specific gene CAR3, showing very distinct features here. You also have ApoA2, which is a very liver specific gene expressed at the liver area. You also have this hemoglobin related genes. So you can see that spreading a lot of places, but they have some hotspot here and here. And this could be, I think later we found out this to be caused by the red cell coagulation, which just the blood cells there. And then we do the call for A1. So this is a collagen spread in endothelial and smooth muscle cells. So we have some like hotspotted regions around this alta area, which just being the endocellular cells. So actins, they are pretty universally expressed everywhere. And we're also doing the spatial DE analysis and can show the heart here, liver here, and lung and dorsal alta here around this region, and also neurotube and lumbar. So we can also label the tissues with those identified features. And another sample we work on is the E12 tail region. So this one we use a lower resolution, the 50 micron, because the E12 mouse is much larger than E11 mouse. So we want to cover the whole tail region, so we have to use a larger channels. Then we can also do a very good identification of heart, liver, lung, and those distinct features as shown here. So then I want to switch a little bit to demonstrate the capability of our technology to do any extra fluorescent labeling here. So basically, if you have a tissue and you want to stain them, for example, we're trying to stain with DBiT, you can see the nucleus, phyloiding, you can see the actins, basically the cell structure or cell skeleton. Then we have another dye here, the P2Y12, basically the stone neurons. So by overlay the imaging here, you can see a very beautiful picture of the mouse embryo. And starting from here, you can use the DBiT-seq data to analyze the spatial area of interest. For example, if you're interested in this air region, what you can do is just put use our PDMS chip to cover this region, and you can enlarge this area and find out the mRNA expression of this region. And another thing you can do, I think, because with DBiT stain, you can see individual nucleus. So that enables you to do cell segmentation. For example, you can clearly see the cells, the cell nucleus, then you can segment them. So this kind of preliminary, we just segment with them using basically the very simple technology to show we can do that, but this can be further improved. And for immunostaining, another thing we can do is we can identify individual pixels. So each pixel will have a pretty well-stained nucleus there. So basically you can count how many cells you have in that pixel. So for example, this one have one single cell. This one also have one single cell, but those two may have two cells or two half cells or more. Basically, you can analyze their gene expression by comparing comparing the cell morphology or cell counting for each pixel, and you can compare the gene expression side by side. So with this, I want to talk a little bit about our technology possibilities in the future. So because we think the banked FFPE samples are pretty valuable for this kind of application, because I think over 90% of the samples in the world has been really banked into the FFPE, the paraffin form, rather than the fresh frozen form. So you can, if we can just access these banked samples, this will greatly push forward our understanding of the samples. Even the FFPE samples are usually considered to have lower quantity mRNAs, but we have done some preliminary data show that they have lower quantity, but they are still good enough to tell a lot of biology from them. So to summarize, I'm just doing a graphic summarize of our technology. So what you start will be a tissue on a slide. Then you can do a H&E staining of it to find, can do the normal annotation or disease diagnosis based on H&E staining. Then you can find the area of interest, then apply our DBiT-seq technology to really focus on that area. Or you can do, if you want to do immunostaining, you can also do that by staining the sample with different immunofluorescent dyes. Then you can also doing the identification of the area of interest. Then you can apply our technology. Then what do you get from this? Our technology will be a gene or protein expression matrix. Basically, you have genes, the individual genes as columns, and you have the spot information, location information, the X and Y for each of the gene. Then what you can do will be, all depends on you, I think. You can do in clustering, you find the differential expressed genes. You can also say individual proteins, or you can cluster based on proteins. Or you can do the spatial identification of more spatial specific tissue features. Another thing you can do is you can just doing a very simple pairwise comparison, either from gene expression or from protein expression. Or you can integrate with a single cell RNA-seq data, which is pretty available nowadays. You can use them to simply annotate your spots. And also you can do more advanced analysis. So I think there are still quite more possibilities there lying. We think there are still a lot of data analysis work being published or being researched these days to get more information from a spatial technology. So with that, I want to thank first my PI, Dr. Rong Fan, from really generating this beautiful idea, and also gave very good guidance of the research for me. And also I want to thank all the Fan lab members, especially Dr. Minyu Yang. She's working hard on the data analysis part of the spatial data we got, and also Dr. Yan Xiangdeng for helping with the fabrication of the PDMS chips. So yeah, with that, I would like to answer any questions you may have. Okay.
Yeah, thank you, Yang. That's a great talk. So any questions, if you have, you can unmute yourself and ask, or you can use the chat box.
Oh.
So Yang, you can read in the chat box some questions there.
Okay, I found a question from Professor Coskun. It's asking what's the ultimate resolution. I think what we do now, the highest resolution we have now is 10 micron, but we do have space between them, is also 10 micron. So I think it's like we have a 10 micron resolution with a 10 micron gap between, and we are also trying to do to increase the resolution to maybe 5 micron or even 2 micron, but that that poses some difficulty there because you have a very narrow channel there. The flowing will be harder because you have much more. You need much more pressure to press the solution through the channels. So we are still working on that, but hopefully we can get some improvement later. Yeah. Thank you for the question. Yeah. Oh, I have another question from Dr. Silas Manitis. Okay, so question is about how reliable the system is, meaning how often do you have problems sealing against tissue or squishing tissue? I think it's a great question. So we do have some of the problems working with the 10 micron resolution channels because for 10 micron resolution channels, we have really thin walls there because the wall is also 10 micron. So the wall becomes thinner, the PDMS will be kind of softer. They're not as firm as a 25 micron or 50 micron. So basically for 50 or 25 micron, it's pretty robust. You don't have any leakage. But for 10 micron, it's a little bit tricky to kind of, it's about how you like clamp it. So you don't want to clamp it too strong. Maybe you can collapse the channel, or you don't want to clamp like too softly, then there could be some leakage. This is something I'm still working on. So basically, you need some experience here to really adjust the pressure or pressure you apply for applied to the tissue. So yeah, there's some trick here for 10 micron, but 25 and 50 are pretty good. Also, another question from Dr. Giura Eger. So asking, why do you speculate you detect 15% of total RNA identified by smFISH? How do you plan to improve it? Okay, so I think we compared it just simply by counts from smFISH and also our counts from the RNA-seq data. We just compare the counts and calculate the ratio. And to improve it, I think some things we can do is first we can improve the chemistry. Basically, we can improve the barcode concentration we apply. That will be a simple way to improve it. Other things we can do is to we can add more. I think add more time for the incubation process because those kind of another thing I forgot to mention, that we can increase sequencing depths. For now, we don't really sequence that deep, so increase the sequence depths may just improve the capture ratio of the comparing with smFISH. So it's a great question. Another question by Dr. Rubin Joyce, and is asking, could this be expanded to a 3D tissue like ISYZ in the future? I think that's doable, but what we are doing now is basically doing a tissue slice. Basically, they are kind of considered as 2D, but you can do is to basically just obtain like tens of them, tens of this kind of slide. Then you can just DBiT-seq one by one. Then later you just overlay them. This is also what other people are doing with other technologies. So by just doing 2D ones, then just aggregate them into a 3D one. I think this is a way we can do a 3D tissue. But if you're asking if we can do the 3D tissue at one time, probably that will be hard for now. We can think about that later. So great question. Okay, another question from Dr. Coskun. Is there any combination pathway? I think we have some really great interest in this method, and they are working on a lot of things, trying to improve the RNA sequencing quality, and also improve basically the liquid handling process, because currently for liquid handling it's kind of hard because there are a lot of injection process. So we have some really talented people working on this to really reduce this, make this process more convenient. Like we can do a liquid injection at once, then flowing once, and possibly reduce the preparation time for the PMS.
Yeah,
that's a great question.
So this year, Ron, maybe I can kind of jump in to answer this question a little bit. I think my lab members just hate to manually pipette 50 different barcode reagents to the device. In particular, we're thinking to expand to 100, so that they refuse to do that. That's basically, I think, some sort of cartridge system can help. And so I think the 3D, in response to the question, so 3D tissue spatial omics, again, indeed, so you can just get a bunch of tissue sections and stack them together. And what is very unique in our approach is we don't need a bunch of tissue sections to do tissue optimization. So in theory, you don't have to waste any precious tissue sections, and it is possible actually for us to reconstruct the entire sort of 3D tissue map with every single tissue section generated from that tissue specimen. And yeah, so basically that is. Okay, I think we are 2 minutes past the 4 o'clock, and thank you so much, Yang, and thank you all for attending the seminar today. And so we really look forward to your participation next week. Thank you all, and take care.
Okay, thank you.
Google is broken.